Welcome, my name is Martin Papst and I would like to explore with you how and why we look at microbes using approaches that go beyond DNA analysis. But let us first take a look at this cell and the different molecules it produces. The genome is the blueprint from which mRNA, proteins and finally metabolites are generated. And while studying the genome with genomics shows only the genetic potential, or the so-called genotype, studying the proteome and metabolome with proteomics and metabolomics reveals the phenotype, which tells us which processes are active and how the cells respond to their environments. And since this course focuses on environmental biotechnology, we will concentrate this module on proteomics and metabolomics of microbial communities. Now let us look at some research questions where you need methods beyond DNA like metaproteomics and metabolomics. For example, you might want to identify which microbes are present in a community and estimate their protein biomass, as this often reflects their metabolic capacity. Secondly, you might want to study which pathways, enzymes, and metabolites are produced under specific reactor environmental conditions. And finally, you might want to study which microbes in a community are the primary users of a certain nutrient resource or the main degrader of pollutants. Well, let us look first at metaproteomics. In short, metaproteomics is the large-scale identification and quantification of proteins from whole microbial communities. This provides information about the microbial biomass, expressed enzymes, and active metabolic pathways under specific conditions. So how does this work? How does metaproteomics work? After sampling the biomass, the microbial sample must be preserved until transferred to the lab for processing. Then the cells are lysed and the proteins are extracted. The proteins are then digested into peptides, which are then separated by chromatography and detected and sequenced by mass spectrometry. Here it is important to note that the mass spec sequencing data need a reference database in order to identify the proteins and the microbial origins. Therefore, metaproteomics is usually performed in combination with whole metagenome sequencing to build a custom reference database that allows to interpret the mass spec data. Finally, the identified peptides and proteins can be linked to metabolic functions and grouped to reconstruct the community taxonomic composition. Here is an example of a metaproteomic metabolic pathway expression map obtained from an anaerobic granular sludge community. On the y-axis are the identified genera, and on the x-axis the key enzymes for nitrogen and phosphorus removal. The colors indicate which microbes express which enzymes or pathways. While many microbes may carry genes for certain reactions, high expression levels are often only observed in specific microbes. For example, the BBK genes are mainly expressed by phosphorus-accumulating organisms like Candidatus acumulibacter. Next, let us look at metabolomics. Metabolomics is the study of all metabolites produced by a cell or specific pathway. However, in microbial communities, most metabolites do not carry taxonomic information, which limits its use in environmental biotechnology. While it still allows to detect certain metabolic conversions within a community, it does not allow to study individual community members. But let's take a closer look to the metabolomics workflow. Generally, there is no single universal method for metabolite detection and analysis. But there are a few key aspects which are essential in any metabolomics study. First, rapid sampling and quenching are critical since most metabolites are unstable and quickly turn over. Second, extraction must be tailored to the sample, as different microbes have different cell structures that need to be disrupted. And third, whenever possible, stable isotope label standards should be included for accurate identification and quantification. Finally, although there is no universal analytical setup, unlike in proteomics, common LC or GC separation methods coupled to mass spectrometry are usually the best choices to achieve sensitive detection of a broad range of metabolites. Well, here are the key takeaways for this module. Metaproteomics and metabolomics reveal the phenotype showing how microbes respond to their environment. And more specifically, metaproteomics tells us which enzymes and pathways are actually expressed. On the other hand, while most metabolites do not provide taxonomic information, targeted metabolite analysis can be very useful for confirming the presence of specific metabolic conversions within a community. For both approaches, however, sample preparation, including preservation, are very critical, and combining them with whole metagenome sequencing is essential to provide biological context. Finally, here I've listed for you a few articles if you would like to dive deeper into the fields of metaproteomics and metabolomics. Thank you.